Hi, my name is Elexuzz Davis, and I go to Alabama State University. This summer, I am working in Dr. Lehrter's lab, and my research is about the reactivity of organic matter in anoxic marine sediments. These photos and videos represent aerobic respiration in a bottle, and the goal is to reach sulfate reduction. Resazurin is used to achieve the color purple and pink, but the clear bottles are the intentional goal of this research. Clear bottles represent sulfate reduction, which means that oxygen is no longer present and the experiment is now in its anoxic stage. My hypothesis is sediments with higher organic matter and temperature will have higher reactivity. Once samples are collected from their sites, along with site water, they are measured and placed into the clear bottles. After collecting data and waiting for each bottle to turn clear, nitrogen bubbling was used to reintroduce oxygen back into the bottles to obtain aerobic respiration. Nitrogen bubbling is conducted to remove oxygen, replace it with nitrogen to achieve anoxia. After nitrogen bubbling, sulfate analysis is conducted. We created a stock and used a barium chloride gelatin reagent to test the amount of sulfate present in our water samples. Because hydrochloric acid has such a distinctive, pungent smell, it is considered a strong acid, so we use this chemical under a fume hood. Afterwards, sodium sulfate powder is measured and added to our stock. This experiment is prevalent because it helps us determine the transition time of aerobic respiration to sulfate reduction. After our trial run for our research, we went out on a boat to collect new sediment samples from the mobile bay. This machine collects the site water from the very bottom. We also collect mud samples and take those back to the lab where we test for sulfate analysis for our research. The mud samples are then collected, drained, and separated by each site. Afterwards, the mud will be weighed one bottle at a time, then site water is poured along with the measured amounts of resazurin with a pipette. The bottles will be shaken and placed in order of the data sheet created to track its process. The importance of this research is sediment organic matter reactivity and how it is related to O2 consumption and hypoxia and the production of CO2 acidification.